WII is a premier institute in India. They are working on several wildlife research and conservation projects. However, they also show us that conservation can happen within the campus itself without spending any extra funds, with the participation of the faculty, their family members, and the students if we follow simple scientific methods. Let us hear the tale of conservation of the Tricarinate Hill Turtle from Dr. R. Suresh Kumar, who is a scientist with Department of Endangered Species Management at Wildlife Institute of India.
So hello everyone. I am going to be sharing a story about a particular turtle. It's a conservation story. And before that, I would like to share with you all a little bit about the conservation issues relating to freshwater turtles and tortoises. So freshwater turtles and tortoises are one of the oldest living group of animals in our planet. And they are faced with the major crisis, which we call the greatest reptile crisis since the demise of the dinosaurs. And this is primarily associated with the massive trade for pet as well as primarily for meat trade. And you know where much of these trade is happening. So turtles have existed on Earth since the rise of the dinosaurs, about 220 million years. And they represent one of the most threatened groups of vertebrates, with nearly 10% of the 317 species that is currently recognized as critically endangered. And some of these species are also functionally extinct, in the sense just a lone individual surviving or a pair which no longer breed, and it's just a question of time before they become extinct. So primarily, almost all freshwater turtles and tortoises are threatened. And when it comes to India, India is one of the mega biodiversity countries in the world, as many of you know. And when it comes to freshwater turtles, within the Indian region, it is again very, very diverse. And it ranks among the top 5 countries in the world in terms of importance for turtle conservation. India is also quite famous, given the fact that the world's greatest turtle species richness area, that is, any one particular region or an area in the world which supports the maximum number of species, happens to be in India and Bangladesh. This is associated with the Ganga and the Brahmaputra river basin drainages, where 18 to 19 species occur within a very, very small area. So it's highlighting to you the importance of this diversity, and that particular diversity is in India. Okay, if you look at this hotspot map, or the global freshwater turtle and diversity tortoise diversity across the world, this is how they're distributed. And coming to India, this red patch that you see is where the highest number of turtle species found anywhere in the world is here in this region with 19 species. So extremely important, extremely unique, and we still know very little about many of these species. So very often people question me, what's a turtle, what's a tortoise? So I thought I will share with you the information on how you differentiate these. And then before we move on, big story about this one particular turtle that we have been researching on. So tortoises are generally dome-shaped, and they have a much larger carapace, and they are primarily terrestrial species. They live on land, and their legs are adapted for, they have elephantine legs because they need to carry the weight and walk around on land. So it looks like an elephant's leg. Tortoises do go into water, but very, very rarely. They do drink water, it's very important, but very rarely. They're primarily associated with arid areas, but they're also found in rainforests. There is in fact one particular species in India which lives in the northeastern parts of India, primarily associated with very dense, humid, wet forests. Now, coming to turtles, generally everybody thinks that turtles mean they live in water. It is true, but that does not necessarily mean that all the turtles, any particular species named as a turtle, lives in water. There are many which live primarily on land and occasionally go to water. So they behave more like a tortoise. And when you talk about turtles, there are two types. One is the hard-shell turtle as shown in this image here, and the other is the soft-shell turtle. So if you look at them, if you look at their overall body plan, the shell is more dorsoventrally flattened, that we say. And this is primarily as an adaptation for living in water. They have a dome-shaped shell, then they would not be hydrodynamic. So as a result, their shell is like flattened. And you would also notice that their legs are modified as flippers, or more, we would just call it as a paddle or an oar. It's not like extended like in the case of the marine turtle. I'm coming there. The freshwater turtles can be hard-shelled or soft-shelled. The freshwater turtles, again, hard-shelled turtles, it does not mean that every turtle will have a webbed feet or a paddle-like or an oar-like. Okay, so this is the difference. There are these freshwater turtles, primarily soft-shell turtles, which are invariably or predominantly aquatic. They stay underwater, they live in water most of the time, but do come out onto the land to bask along the river banks to bask, or also to lay their eggs. So all the species have to come out of water to lay the eggs. Okay, so moving on to the marine turtles. Marine turtles represent the other extreme of living in an aquatic environment or in a marine environment. They spend all their life moving around in the vast oceans, and so their four limbs are modified into what we call as the flipper. So again, dorsoventrally flattened shell, and that's how you differentiate. And they primarily remain in marine environment. Very, very rarely they come into the aquatic environment. So with that brief intro to you, I move on to the story about this particular freshwater turtle, which is known as the Tricarinate Hill Turtle. We knew very little about the species, and over the years, through a long-term monitoring project, we have managed to gather some very important insights about this very, very interesting and unique species. So it's called the, it's basically, I've titled it as The Life of the Tricarinate Hill Turtle, and I will tell you about the species. Now, the story that I'm sharing with you here is all published in a paper and in an international journal called the Herpetological Journal, published by the British Herpetological Society. And since this particular paper was dealing with this species for the first time, providing insights about its ecology for the first time, they actually featured it on their cover page. So the story about this particular turtle is this. So I had told you that there are these freshwater turtles. They are otherwise also known as semi-aquatic pond turtles. And the species that we are dealing with are known as Old World pond turtles. Old World refers to the continents other than North America and South America. So this particular Tricarinate hill turtle belongs to this family known as Geomyidae. Geomyidae in India, we have 16 species, and 2 of this species belongs to this particular genera known as Melanocheles. Melanocheles, melano means black or lack of pigmentation. Cheles means the shell. So 2 species, like I had told you, the Tricarinate hill turtle, about which I'm going to be speaking to you about, and the Indian black turtle. So the Indian black turtle is a widely occurring species, is found across India, across the Indian subcontinent, Nepal, Bangladesh, Bhutan, as well as the whole of India. Quite a generalist species, and occurs in forested habitats as well as in agricultural landscapes too, wherever there is water that is available. On the other hand, the Tricarinate hill turtle is distributed along a very narrow strip along the Himalayan foothills. Okay, and this particular species, the Tricarinate hill turtle, Of course, I told you the hill is referring to the foothills along the Himalayas, but also you notice on the carapace or on the shell, there are these three lines, three orange yellowish marking. Those lines are known as keels or, carina, and that's why it's called Tricarinate Hill Turtle. Now, this particular species is also associated with the sal forest. The sal is a hardwood tree, Shoria robusta, found all along the Himalayan foothills as you see in this map. But the sal forest distribution is also there in some parts of eastern India, in central India. And you see the star there, that is the type locality. This is a location in Jharkhand. That is where this particular species was first described from in the 1800s. And anything more to say? This is a species listed under Schedule 1 of the Indian Wildlife Protection Act. So it's a very, very serious offense if you happen to have this particular turtle. And it is listed under the Appendix 1 of the CITES, which is a convention that governs trade in endangered species. So it is under Appendix 1, which means that no trade in this particular species is allowed. And under the IUCN Red List assessment, okay, so all of these, so this species is well protected in at least on paper, but how much do we know about it? So when we started this work, there was very little information in literature that is there, and hardly anyone knew about where all is this found. Is this species found widely distributed as shown here in this map along all of those green areas? It's still not known. Okay, so thankfully for us, this Tricarinate Hill Turtle occurs within the Wildlife Institute of India campus, as shown here in this slide. And you see those green forested areas. This is where it is. Much of these areas have been preserved by our former directors with the vision that we should have this as our backyard research area. And with protection, the forests have grown back and the turtle populations do survive in this. And that's how we stumbled upon them and came up with the idea of monitoring these populations over time. So the WI campus is about 32 hectares, and adjoining to this is another large patch of forest, which is again kind of connected. So there is some amount of inflow of turtles from there into the campus, and possibly some of our turtles do go outside. And very important aspect about Dehradun is also that we face both hot summers as well as winters. Now, cold winters is something a challenge for reptiles because turtles or reptiles are cold-blooded, and in very harsh cold weather conditions, they find it very, very difficult to survive. So when we initiated the study, or when we were planning on the study, we were curious about what do these turtles do when it is very cold here in Dehradun. And the other very important feature about Dehradun is also that it has a very unique climate. We receive about 2000 mm rainfall, and much of this within a span of 2 or 3 months during the southwest monsoon. So a lot of rain in this area. So these are a few pictures of the campus, the natural part of the campus. There's good amount of a lot of leaf litter and predominantly sal-dominated forest. So when we started this work, started off with simple walk along the existing trails looking for turtles, and when we chanced on a turtle, we used to bring it to the office and take some measurements. That's what most of us do when we are researching a particular species. Now, before I proceed with the story, we're dealing with the Schedule 1 species, and it's important that we respect the law. So we had taken prior permission to undertake this study, even though it is within the Wildlife Institute campus, and with permission from the Uttarakhand State Forest Department, we had initiated this long-term monitoring, and we keep reporting to them about the progress of this work. So turtles were, so when you chance on a turtle, the first thing that you would like to know is whether it's a male, female, what is the size of this turtle? So there are set standard measurements that we take. So for example, here in the slide, you'll notice that we are measuring the length of the shell. These are measures to tell you about how old these turtles are, or with age, the shell or the body size does grow. So in order to monitor all these, we have to take these measurements. So the carapace, and then you have the plastron. In the case of this particular turtle, in the case of a male, the plastron, as you see in the image, which is basically the underside, the shell on the underside, is concave. There is a cavity. So that's how you differentiate that this is a male. In the female, you wouldn't have that concavity or the cavity there. So it's quite easy to distinguish in the field when you pick this up. But again, you can only distinguish whether it's a male or female only after a certain size class or after an age class. Prior to that, both the males and females, because the young turtles wouldn't show this concavity, you would not be able to distinguish the sex. Okay, so weight is another very important parameter to record, because males could weigh more than females or females could weigh more than males, so needed to know these measurements. Thankfully for us, this particular turtle also had these unique growth ring-like patterns on the shell. You notice the shell, it's actually composed of different shell plates. So there are shell in the middle line, those are the central scutes, we call it. The ones on the side are lateral, and along the edges, that's known as the marginal scutes. So we can actually see these lines in each of these scutes, and it's supposed that, it's assumed that each of these lines do indicate a year of growth. So a simple count of these rings can actually help you understand what is the age of this particular turtle, because many people think that turtles live very, very long. It's not necessarily true that they live very long. I'll come to that story. So over years we've found that these turtles survive for about 15 to 17 years and not more. So that is the story about the longevity of these turtles. And now moving on further to what else do we do? So we note down the location, then we collect dropping. Very often when we pick up turtles, they poop as a strategy, defense mechanism. So we collect the poop. By looking at the poop, you can actually know what they're actually feeding on. So collection of the poop was very important. The morphometric measurement that I just told you, age determination. Then finally, before we release, we mark them, because there's a very high probability you may be ending up catching the same turtle. If you haven't marked, how would you know that this is the turtle that we had caught a few days ago or a month ago or a year ago? So each of these turtles were uniquely marked, and that's where I'm coming to. So there are different marking systems that turtle biologists follow. And we have tried to adopt some of these measures, and we have to suit our needs. So if you notice here, what I have shown in the diagram is basically the marginal scutes, which can be actually used for marking individual turtles or uniquely marking individual turtles. So it's a combination of these scutes, and what you notice there is on the image is the scute notch. By filing a small notch in the shell, and towards the edge of the shell, it's more like your nail. And if you do make a cut there, it does not actually cause any harm to the turtle. So, and these marks remain for life. So this way, if you're planning on marking turtles for, say, 20 years, or if your program continues on, and if these turtles survive in the population, you would know that this is that particular individual. So every individual that we captured on campus, we mark them and release them back in the same spot where we picked them. So that's very, very important. I'll come to that story later. In literature, there was not much mentioned about the sexual dimorphism. Of course, I earlier told you that the plastron concavity in the males is a distinguishing mark that it's a male or a female. But over time, when we started collecting data, we noticed that on the right side, you notice there is the female, and above the eye on the forehead, there is this stripe. There's a pink stripe on the brow, which we call the superciliary stripe. Now, as young turtles, both male and female, will have the superciliary stripe. But as the young turtle matures into an adult male, it will start developing the concavity, and at the same time, it loses this pink stripe. And during breeding season, this particular pink stripe, in the case of females, become more prominent. It becomes more pinkish. basically indicating its reproductive state or status. Okay, so why the males don't have, why they still something that we don't understand, but this is something which was not reported in literature, and we happen to find this. So I told you about the poop. Oh, it's a wealth of data that we could generate from just cleaning the poop, washing them and looking under the microscope, but just looking under a hand lens. There were so many things that these turtles were feeding on. They seem to be omnivorous, but they point of time feed entirely on one particular plant here known as the jasminum. It's the wild jasminum, Jasminum multiflorum, the fruits of it. In fact, at this point of time, the plant is flowering, and in about a month's time, it will start fruiting. And so when the first shower of the monsoon arrives, the fruits ripen. And that's when the turtles are going to be active, and they're going to be actively seeking for this particular fruit that they will consume. It has a very, very little epicarp that's not much fleshy in terms of, but some of these turtles survive just feeding on that. And of course, there are so many other interesting things that they fed on. Earthworms is another major diet story. I will come back to that. So if you look at this, the jasminum composed of 52% of the diet, other fruits, Cordia dichotoma, then animal remains like the earthworms. Unfortunately, in the poop, most often animal remains, which are basically soft tissue, is all digested. So you are probably biased in your reporting of animal remains, but it tells you that they're fairly omnivorous in nature. So moving on to other things that we found about this particular turtle, that the first and foremost is that most of our captures were seasonal in nature. Not that we were not walking around looking for this turtle in the other times of the year, but somehow there was a rainfall connection to it. It appears like rainfall excites these turtles, and that's when they're moving around everywhere. So the moment it starts to rain, and if you go out into the forest or out into the nature part of our WI campus, the chances are, the probability of you encountering a turtle is very, very high. So this figure here, you can actually see maximum captures is associated with peak rainfall in the area. That is in the month of July, when when the southwest monsoon is at its peak. So there's a very strong correlation with rainfall. But post that, your captures decline. And in the winter months, which is starting from November all the way into February, there's hardly any capture, indicating that these turtles possibly go and hide somewhere in the forest floor. So I'll come to that story a little later when we discuss about the movements. Now, what does all the size measurements that we were taking telling us? It basically tells you about the population characteristics of, it basically characterizes the population on the campus, on the Wildlife Institute campus. What you can actually see here is that most of the turtles that we caught were primarily adults, or the maximum captures, it was in the range of about 67 to 70%. Most of it is in the size range of 130 to 170 or 180 millimeters. Okay, and what we also notice here is that the females are smaller. By the red bar that you notice, the females are smaller, while the males are much larger. Okay, I will tell you more stories about that a little later. Now, if you come to the size class somewhere between 50 to about 100, there the captures are generally very, very low. So there are two possibilities. One, at this particular size class, there could be very high mortality happening, or it is just that there is a capture bias. We are not able to detect them that easily as the large turtles. Now, why do we see more number of hatchlings? High chance that you could be, the hatchlings could be, getting killed or getting eaten up by predators. But it is just that during a very short period of time, you happen to see more of these hatchlings. So when you pull in all the data, it shows that there is a lot more hatchlings in the area. But it is the same hatchlings that are here that actually get recruited as juveniles, then subadults, and then as adults. So there is definitely a capture bias in the sense that you tend to see more larger individuals. So it's interesting as to what is going on with these young guys. So we need to track them too. Now, it's a bit complicated, this graph, but it's not all that complicated. We have basically regressed the carapace length, which is an indicator of size, versus weight. Okay, and when we regress this for the males and the females in the population, what we notice here is that the males tend to grow longer, and as the larger they are, the more heavier they are. Okay, while the females are much smaller. Now, what this basically indicates to you is that, and the females are also on the higher side here, as you notice. At this size class, they are weighing more. At this size class, the males are weighing less. So the females are maturing much earlier. And after a certain point of time, they do not grow. While the males continue to grow and they mature only later on. So it's only these older population get to mate with these females. So this particular aspect is very interesting because this is a tortoise character. Most or almost all tortoises follow this principle. Whereas the aquatic turtles, or those species that are primarily aquatic, freshwater turtles, hard-shelled, there the size is reversed in the sense the males are much smaller while the females are much larger. So this particular Tricarinate hill turtle, even though we call it a turtle, it is primarily terrestrial in nature. That's what we find. And the other aspect of it, it shows more tortoise characters, which is evident from this figure. Okay. So we also took measurements of the age and counted the growth rings. And what we notice is that at a certain age class, so somewhere between 10 years to 8 to 10 years, beyond which it becomes extremely difficult for you to enumerate or count the number of growth rings that are there because they're very, very close. And it's also that after putting on 10 rings, they may not add any more rings, but continue to remain in the population. So the graph the lines stabilize here at the age of 10 to 12 years. It's not, you cannot easily distinguish. Okay. So now from there, moving on to the movement ecology study, which is one of the most interesting aspects because you would like to know where all these turtles move around. But what I would like to share with you all here is that during our efforts to locate turtles, we went to the same turtle that we had marked in the same spot. So over years, you recapture that individual and you come up and you find that this individual invariably is caught in that same particular spot, indicates that this shows high site fertility. And this was primarily the case with females. So the males could possibly be moving around, but there was the story about site fertility. So it was very interesting, or rather was quite important to document what is the home range, how much do they move, where all do they move, and along with it, you also get information on activity pattern. Are they active during the day? Are they active during the night? Are they active across the year? Or they're active only during a certain time of the year? All of these stories. Okay. So I'm going to be telling you about the tracking aspect of the study, where invariably when we talk about tracking, we think of certain devices or instruments. You will like to instrument your animal, you put a collar, you put a tag, you attach it, and you track them. Well, all of these devices are available, but they all are expensive, relatively. Okay, not very expensive, but there are also very expensive units. Now, each of them come with their own errors. So, get into that finer details because it's very lengthy and long. But I am going to just share with you the story of what we actually did. Now, in this image that you see here, there is a gully. The side, the terrain is a little more steep, so would not move on to very steep terrain, so that the gradient of the slope is very, very important. So you would probably see turtles negotiating these using select paths in the area. And it becomes very important to actually map the exact path in which they have moved around. And for that, you need to have devices that can precisely measure every few hours or few minutes, and that's going to be a lot of data, and it's going to be very time consuming to and there are other challenges associated with those kind of instruments. So how else can we actually track these turtles? Okay, so there's a lot of researchers around the world who have used very simple techniques way back in the prior to the 1950s by putting a simple spool of thread on a turtle to actually monitor its movements. So this is known as the thread trailing method. And so we came up with this idea of looking for trailing these individuals, turtles in our campus. And here what you see in this image is a tracking bobbin. It's a specific specialized one. You actually see that it doesn't have a cardboard. Generally, when you look at a thread bobbin, you will see a cardboard, but this one doesn't have. This is specifically made for some specific purposes, and this was imported from Germany. And each of these units are going to cost me $2 and at that point of time it was about 90 rupees, but today it's much more than 90. It's about close to 180 rupees. And the tracking, the thread lengths were only about 250 meters, okay, and they weighed about 6 grams. Now the length was the biggest issue for me, in the sense that if you put the 250 meter thread and if your turtle happens to move 500, the turtle is gone. And to be turtle every now and to keep changing the spool. So your researchers influence handling the animal is going to influence its natural movement or natural behavior. So you need to come up with something which is ideal, where there is less handling, and at the same time it captures the data of where all this particular turtle moves. And of course, the cost was also something at that point of time prohibitive. It was about 180 rupees at that point of time. This is a story I'm talking about nearly 15 years back. And so we came up with a simple thread spool that you just go to your shop nearby and a tailoring shop and just ask for a thread. And I chanced on this that there is this Madura Coats cotton thread, which is about 800 meters and weighs 24 grams. And it costs just 8 rupees. But the thing is, it comes with the cardboard. How do you remove the cardboard without actually, changing the shape of the spool? Now, before I go to that, I'll tell you one more story, or an important aspect that we have to consider when we track animals, can be any animal. The tracking unit, whether it's a device or a thread spool, has to be less than 3% of its body weight. In mammals, to 5%, less than 5% of its body weight. Anything more than that, you would be altering the natural behavior because the animal is carrying it and it has to lug around. Okay, so there is an animal ethics that you need to follow, and it has to be less than 3%. And this particular cotton thread for 8 rupees did meet the criteria of being less than 3% of its body weight that we could use in this turtle. So we somehow figured out that, if you just put the spool between your molars or premolar and just gradually pull out the cardboard, it does come out. The intention of pulling this out is that you need the inner end of the thread, which needs to be tied to a branch, and this whole spool is put onto it. So as the turtle moves with the spool, the inner end of the thread keeps coming out. So that's how you trail. If you take the outer end of the thread, then the spool will have, or the bobbin will have to spin. So that's a problem. Okay, so that's how, it's a very simple technique, and you can see it, it's taped onto the back of the turtle, and you tie the free end to a small branch or a small tree or something like that and let go of the turtle where you captured. And for an initial few minutes, the turtle might feel there's something on it, but over time it gets over it, and you actually see the turtle leaving a trail behind, and you get to know where all it is moving around. Like I told you earlier, there are a lot of researchers who've done different methods, who use these thread bobbins or thread spools to track the movements. And I'll mention to you, way back in 1927, there's this guy by name Breder who had used this particular technique. Amazing. You cannot ask for a better information than what the thread trailing technique actually gives you, because it tells you exactly where it went. So over time, we tagged quite a few individuals, and we tracked them. And yeah, we tracked them for really long. One particular individual, close to a year, we tracked this particular turtle, and we measured the track. It was almost close to 5,000 meters, or 4,290 meters to be precise. And I had also told you earlier that it also gives us information on activity. And turtle remained 3 months inactive at one. Okay, so this is the story that we were really looking for. What do they do? Which time of the year that they're inactive? And naturally during winter that they're inactive. So I'm going to be sharing with you a few images here about the turtles on the campus. So to try and recollect the earlier image of the WII campus, all of these areas that you see here, these is all the forested patches, and there is a lake associated with this area. And this is where a couple of turtles were being tracked. And you can see the green one is an individual which we named female number 1. In fact, we had tagged marked way back in 1998, and it was then female number 22. So they did seem to have an overlap, but they had their different areas. There is female number 18, and then a female that came from that was released on campus. Finally, there's this another female that just showed you. Now here, this is interesting. Now you see all of these females are all overlapping one another. But this male, which we had marked it as 39, he actually overlapped all of these females. And he was the bull turtle at that point of time. So he's probably passing on his genes to all of these females. He probably had, he was probably maintaining something like a harem. Of course, there are other bull turtles in the campus, and there's possibly male-male competition that goes on. And it's very difficult to track males because they ranged very far and wide within a short period of time. So, so in fact, our data sets are primarily centered around the female because we could track them for long, mainly because they were confined to small areas. Now, when I say that confined to small areas, recollect the story of the site fidelity that I was telling you. And also here it is important to mention that, you know, very often somebody goes and picks up a turtle and then they go back and release it. They would just go and drop it elsewhere. You know, you're going to be completely putting it in a completely new area where it is not familiar with. So when you see this thousands of turtles that are being traded, that are being seized, or that were originally a consignment that was heading out of India or to West Bengal or to any of the other states where there is consumption still, the forest department do seize them. Where do you release them? Just go and release them in some river or in some forest or something like that. You're actually messing up with their knowledge about their area that they live in. So it's very, very important. So even within the Wildlife Institute of India campus, very often someone would bring a turtle, and I would be very particular about where exactly they picked up this turtle, because they need to go back and release it there. Otherwise, that particular individual is lost even within the Wildlife Institute campus. So the other aspect that I'm going to cover here is about the winter and summer range. Look at this female number 22. This is where it moved around during, but during winter, the home range reduced to a very small area. From nearly 11,000 square meters, it reduced to about 1,200 square meters. So you can see that during winter, they become very, very localized and inactive during that period. Similar was the case with another female, very similar in terms of the area that they occupy. And so this particular female, even in summer, ranged over much smaller area, only about 5,700 odd square meter. And in winter, it was very relatively much lower. Similar is with another particular turtle. Now, when we look at the movement pattern and we compare it with the rainfall in the area, if you recollect again the rainfall, which is an important factor governing the movement of these turtles, clearly you see during the non-rainy months, November, December, January, February, they are completely inactive. And you can also see there is the temperature, there is a dip in the temperature, it becomes very cold. And so turtles, being reptiles, need to conserve their energy. So they possibly may be going into a state of hibernation. Okay, our understanding of this is still very limited, whether they truly hibernate or they just go into a state of inactivity. Okay. So the other important aspect that you see in the red letters below is about the number of days that they remained inactive. This particular individual remained inactive for nearly 4 months. So in fact, even during the other times of the year, that is, post-winter, sometime in spring or post-monsoon, that's from September onwards, before the winter actually arrives, even their movement is very less as compared to the movement during the monsoon months. So in fact, to sum it up, these turtles are actually active only for 4 months in a year. The rest of the year, they remain inactive or with very minimal movement. And this is all governed by what is the food that is available to them. So just before the monsoon arrives, I had told you that the jasminum or the wild jasmine fruits are available. But when it starts to peak, when the monsoon starts to peak, that particular fruit disappears. You do not have that fruit any longer. So, but as the monsoon starts to peak, that is when earthworms emerge. And the turtles quickly switch to an animal diet or a diet primarily of earthworms. So this particular life of this Tricarinate turtle is, is all under the influence of the rainfall. So it becomes very interesting what happens in years when there is very poor rainfall. It's a drought year. There's not much rainfall. Maybe this is influencing the fruit production, the jasmine fruit production. Maybe this is influencing how the earthworms are behaving in terms of their breeding and emergence from the soil. So in fact, this is again a fantastic subject for long-term impacts associated with climate change. So if you're looking at the flowering, the phenological aspects of the jasmine, which is one of the key plant species or diet of the Tricarinate hill turtles, and again the earthworm. So does this all translate into whether they reproduce at all that particular year? And even if they do reproduce, and even if there are hatchlings that emerge, is there enough food available to them? Do they survive? Do they make it? So all of these are very interesting questions, comes through this very simple monitoring, but of course, this needs very long-term data sets to understand that. Okay, so thank you for listening. This work was not done just by me. There were a whole lot of individuals who are supporting this. It's more like a campus biodiversity monitoring program, and there's a turtle monitoring program under that. There are a lot of enthusiastic individuals, students helping us monitor this. The Uttarakhand State Forest Department, like I mentioned to you earlier, they're responsible for giving permissions for the study. And there's a whole lot of people that I'll have to acknowledge. A lot more stories, but I'll stop here. Thank you very much.